Oh, hello everyone. I'm Ananth. I'm a third year PhD student based at the MRC Laboratory of Molecular Biology. So before I go into my talk, I'll say a few words about what I've done before I've come to Cambridge. So I did my undergrad in engineering in India, and in my final year of undergrad, I was getting more interested into basic fundamental science. So I did an internship in Okinawa studying the bacterial flagella motor. And ever since then, I've been interested in studying multi-protein machineries. And I did a master's in Nagoya University, again studying the bacterial flagella motor. And that's where I got fascinated by structural biology. And that led me to the LMB, where I'm still studying biomolecular machines in the cell, but I'm not studying the flagella motor anymore. Instead, I'm studying another machinery called the mRNA 3' processing machinery. So in the next 20 minutes, I'm going to talk about the work I've done in the past 2.5 years at the LMB. So I really like this picture. This picture is that of a cross-section of a human cell. This is an electron micrograph. And for those of you who remember your high school biology, you might know that a cell has a nucleus and a cytoplasm separated by nuclear membranes. And the picture on the right actually animates and colors some of the features that you can see, which you can't clearly see here. And the reason why I'm showing this image here is because I want to highlight the importance of molecules such as proteins, DNA and RNA, which form like the building block of cell, which in turn forms the building block of all life forms. So here shown in yellow are the nuclear density, which is majorly made up of DNA, RNA, the nucleic acids, and these rings here are the nuclear pore complexes through which the DNA, RNA in the nucleus can enter the cytoplasm. And cytoplasm can be thought of a bag of a soup of chemicals which has a lot of proteins and RNA mixed together doing a lot of functions. And another thing I wanted to highlight was the cell can be thought of a huge factory made up of tiny machines. And for example, these yellow dots here are a part of a machine called the ribosome, and ribosomes are the protein synthesis machinery in the cell. So in the next few minutes, I'm going to be introducing a machine that I'm interested in, and my research is about understanding how that machine works. And before that, I would highlight the central dogma of molecular biology, where the DNA present inside the cell gets transcribed into another molecule called the RNA, and this RNA is then translated into the proteins. So these three steps forms the basis of almost all life forms on Earth. And the DNA and RNA are made inside the nucleus, and then from the nucleus they are transported into the cytoplasm where proteins are made. And my research primarily revolves around this fellow here called the RNA and the modifications that he undergoes in order to carry out its function. So coming back to the nucleus and cytoplasm again, so here in gray is the DNA, and you have a molecular machine called the RNA polymerase 2 that faithfully makes a RNA molecule from the DNA. And this RNA molecule, as it's being made by this polymerase, is called a pre-mRNA. M here stands for messenger, and the reason why it's called messenger is because it takes the message from a DNA, it goes all the way out into the cytoplasm, and it delivers the message to the ribosome, which uses the information that the DNA initially gave to make proteins. So I often give an analogy where you have a rare textbook in the library, which, there's just one copy of the book, which is the DNA, and a student wants to make some notes from the book, and the student is the ribosome, and the librarian would not give the single rare copy to the student. He or she is going to make a photocopy of that rare book, and that photocopy here is the RNA. The librarian then hands the photocopy or the RNA to the ribosome or the student. So I really like this analogy. So coming back to the pre-mRNA, which is what I'm interested in, it undergoes several modifications in the course of its life. So you might remember this picture from my first slide. It's the picture of a medieval messenger delivering a message from his master to the recipient of the message. So messenger RNA does something very similar. It takes the message from the DNA and takes it to the cytoplasm and delivers it to the ribosome. Let's take a further look into the messenger. So he has a tunic, he has some provisions, he has shoes and a pouch. Similarly, the messenger RNA has a cap in one end. It undergoes some processing called splicing in the middle, and in the other end called the 3' end, it has few other accessories. One such accessory is called a PolyA tail. So it's basically a repetitive addition of adenosine. So this is the accessory or the modification that I am primarily interested in. Well, you might ask me, why should I be bothered about the PolyA tail? Well, I would argue that the PolyA tail is important in order for this messenger RNA to be exported into the cytoplasm. And once they go into the cytoplasm, they are translated by this molecular machine called the ribosome into these protein molecules. And PolyA tails are also important for this process. And those mRNAs which do not take this route take another route where there are few other molecular machines in the cell which deadenylates, or in other words prepares the RNA for degradation. And turns out that the longer the PolyA tail, the more stable the mRNA is in the cytoplasm. So PolyA tail also confers stability to the mRNA. So these are some of the basic functions of PolyA tail, and that's why it's important to know how they are added and why they exist. Let's take a look at a few of the other molecular machines in the cell. I spoke about the polymerase II, and then the spliceosomes and the ribosome. There are several high resolution 3D structures available for these molecular machines. And we need such high resolution or atomic resolution 3D structures because only if we can see those protein complexes and understand how they assemble and interact with each other, we can get some idea of how they function. But if we look at the molecular machine known as CPF, which is called the Cleavage and Polyadenylation Factor, which carries out the process of cleavage and polyadenylation, there are no such structures available so far. And there are also not many biochemical studies based on the structures. There are structures of the individual components that make up this complex, but there is not a structure of the whole complex together. So Ashley Easter, a PhD student in the lab, he managed to purify the entire CPF from yeast cells. So this here is an SDS page gel. In simple terms, every band that you see in this gel represents a single protein molecule, and as one can see, there are 14 molecules here, and there are no other bands in between the ones marked here, which means there are no unknown bands or degradation bands. So Ashley managed to isolate this complex CPF from yeast cells, and it was very pure. And Ashley's work also told us about the protein-protein interaction network within the complex. So CPF is a protein complex with 14 different proteins, and he was able to show which protein interacts with which other protein and how they are arranged. But however, we still do not have a molecular mechanism of how the messenger RNA is cleaved and it's being polyadenylated. And in order to do so, we need high resolution 3D structures. And when I joined the lab, I was interested in the process of polyadenylation and how the PolyA tails were added. Turns out that this 5-protein complex called the polymerase module, which is like a subset of the CPF, that's enough to add specific PolyA tails to an mRNA. So I chose to focus not on the full complex, but just a module of the complex. Let me say a few more words about this module I chose to work with. So it's called the PolyA polymerase module because it has the polymerase PAP1. So PAP1 is the enzyme that adds the polyadenosine to an mRNA. And PAP1 is associated with few other proteins, and one of them is CFT1. It is a protein with high beta strand, and there are two other proteins which binds RNA, and then you have a totally unstructured protein called FIP. And these proteins are from yeast, and humans also have analogous proteins in them. What really interested me was this PAP1 enzyme alone can add a PolyA tail to an mRNA. In such a scenario, I was really fascinated and curious about the existence of PAP1 along with few other proteins. So I wanted to know what were the functions of the other proteins. And in the context of this whole complex, how does RNA bind, and how can PAP1 access the free end of an RNA to add PolyA tails? So we chose to take a biochemistry and structural biology approach to answer these questions. So I said I wanted to work with this complex called polymerase complex. I first have to make the complex physically in my lab in the test tube. So I make use of this technique called baculovirus mediated insect cell overexpression. I can break it down for you guys. So these proteins they have genes that encode for these proteins. If you remember the central dogma, every DNA will have information to make a particular protein. So this protein PAP1 will have a gene that encodes for that protein. So what I did was, I took the genes from yeast that encodes for these proteins, I cloned them into another DNA which was used to generate a virus. So to make things simple, I am using a virus which will produce these proteins for me once the virus infects an insect cell. So basically, I'm making use of a virus to infect an insect cell and make the insect cell produce the protein for me. So I'm hijacking the insect cell. At the end of the day, I get my proteins. So this is again an SDS page, and polymerase module has 5 proteins. You can see 5 bands here corresponding for each of the subunits. I was also able to make a protein that does not have the polymerase PAP1. So I'm going to spend a few minutes explaining this gel to you. This is a polyacrylamide gel. So once I made this proteins in my lab in the test tube, I wanted to make sure that the protein actually does its function, because I'm not taking it from its native source. I am recombinantly producing it. So I want to make sure if it's functional. So this band here is an RNA band, and this RNA can be acted upon by this protein complex in the presence of ATP, and this protein can add a PolyA tail to the RNA. So this is a 2-dimensional gel. The RNA starts migrating from the top of the gel, and you apply a potential across the gel. So the smaller the RNA, the faster it migrates. So this RNA goes all the way to the bottom of the gel because it's only 42 base pair in length. Whereas if you look at these RNAs, they're still on the top of the gel because they are longer. And they're longer because this RNA, when mixed with this complex, ends up getting a PolyA tail or a tail at its end. So what this gel tells me is the proteins I've made are actually functionally active. So now I have recombinantly produced PolyA module in the test tube in my lab. The next thing I wanted to do was to understand how this protein can add a PolyA tail to the RNA. As I mentioned before, we take a structural biology approach. So there is a very interesting technique called electron cryo microscopy, which won the Nobel Prize in chemistry last year. One of the co-recipients of the Nobel Prize is based at the LMB. So we were very fortunate to have such advanced techniques at a very early stage in the field to be accessible to us. And this really accelerated the progress of the project. So I can quickly go through how this technique works. So I have purified the protein over here. I want to know how the protein looks like. I literally want to see the protein, and that's why I use an electron microscope. But I can't put the protein directly into the microscope chamber because you have very high vacuum. I need stable supports like the one shown here. So these are tiny circular discs that has tiny meshes in them, and these are made of gold. What you do is you take your protein and put them on top of the grids, and then you freeze them in liquid ethane very rapidly at minus 189 degrees Celsius. And this frozen sample is then introduced into the chamber of a microscope. This is one of the microscopes that we have at the LMB. It's a 300 kilovolt electron microscope. And then you get these kind of images here, and you have sophisticated computer programs which can then calculate the density map of your protein molecules. And then at the end of the day, once you have a very high resolution density map, you can build your protein molecules into the map. So this is a rapid crash course on cryo-EM. So these things here are individual protein molecules randomly suspended in buffers, and then you flash freeze them in minus 180 in liquid ethane. So now you have these protein molecules in random orientations in the buffer, and then you shoot them with electrons, and then you have a detector which can receive the scattered electrons, and these boxes contain images of the single protein molecule. So these are basically 2D projections of the proteins in ice. Now we are trying to calculate what orientation of these 2D projections corresponds to the three-dimensional object. And we randomly assign orientations to the 2D projections, and then try to iteratively figure out which would be the right angles. And once we determine the exact angles, we do something called a 3D back projections. And now you can get a confident three-dimensional model. To make things much more simpler, it's very analogous to a 3D holography. But basically at the end of the day, you get atomic resolution structures of biomolecules. So Anna Kesenial, a postdoc in the lab, and myself, we collected cryo-electron microscopy data both at the MRC LMB in Cambridge and also at Diamond in Oxford. We both managed to get some nice images of the PolyA module or PAM. So these black dots here represent single molecule of the protein. So let's look at this molecule here. All you can see is just a single dot, and that's because biomolecules are susceptible to radiation damage. So we shoot them with a low dose of electrons, but we need to get high resolution information. Here's where clever image processing comes into the picture. I extract this particle from this image, and I superimpose that on top of thousands of such same orientation extracted particles and stack them on top of each other and get something called 2D classes. And these 2D classes now have a very high signal to noise ratio, and now you can see very fine detail features. So here you can see helices, and once you see such very fine details in these 2D classes, you can now confidently recombine in three-dimensional space to get a three-dimensional map of the atoms in the molecules. So we were able to get a 3D map at 3.4 angstrom resolution, but as we go away from the center, the resolution goes a bit low, up to 4.5 angstroms, but still this is considered really high resolution in structural biology because you can start seeing separation in the secondary structures of amino acids, and amino acids are the building blocks of proteins. And this was the density map of the protein complex I purified. And the problem now was to identify which part of the map belong to which protein, because as of now you just have a scaffold of the whole protein. But luckily we know the primary amino acid sequence of the individual proteins that we put onto the microscope sample. So I can give you another analogy here. Let's say we take a cat and we cover it with plaster of Paris and then make a scaffold of the cat. There's no way you can put me into the scaffold that you made from the cat. Something very similar happens here. So, you know, these big aromatic amino acid side chains cannot be fitted into a smaller density region here. So you manually fit the density map we obtain based on the primary sequence information you know about the proteins. At the end of the day, you get something like this. We call it a model for the protein complex. So we were able to build a model for the PolyA module because we had very high resolution density map, and we were able to get a lot of information which was previously unknown because there were no high resolution structures. One of the most striking thing was this model we built for PAM here was very similar to several other nucleic acid binding protein complexes which had already been solved and modeled. So this is a complex called DDB1, DDB2. You might now know that this complex PAM adds polyA tail to an mRNA. This complex DDB1, DDB2 is involved in repairing damaged DNA, especially UV light damaged DNA. And this is another protein complex which has a very similar architecture to that of my complex, thus showing that there is some sort of evolutionary relationship between these nucleic acid complexes. I must also say that the structure of this complex had a DNA bound to it. So this is a cartoon representation of a DNA molecule. But however, we don't have any nucleic acid bound to my structure. So that's where structural alignments came into rescue. So there are several molecules of amino acid in the top region of this complex which interacts with DNA. In my structure, we found very similar molecules which are highlighted here, which has the potential to interact with a negatively charged moiety like a nucleic acid. So comparing my structure with a previous structure, we can now predict how this protein complex can also interact with nucleic acids. And I also verified their interaction biochemically. I'm not going into the details over here. So I'm going to talk a bit about this technique called cross-linking mass spectrometry. So I told you that we have modeled amino acid sequences into a three-dimensional map we obtained. We have to validate if our modeling is true or not. So what we do is we do a bunch of biochemical experiments which can give you information about proximity of amino acids in solution. Let's say I am a protein and I have an amino acid phenylalanine in this part of the three-dimensional space, and there is another amino acid in this part of the three-dimensional space. And in my model, if I had built them closer, biochemically I can get information about how far they are and how close they are. To be more specific, the biochemical experiments would give me information about a particular amino acid called lysine. So it's a bit hard to explain it to a non-biology audience, but you got to have my word on this. We were able to verify the 3D model we built because some of these arrows we show here actually correlated well with the biochemical mass spectroscopy we did. So one thing I have to mention is this PolyA module complex has 5 proteins, but the Cryo-EM model we have has only 3 proteins in them. That's because The other two proteins are disordered, and it's really hard to see them in the microscope images. It's actually as simple as this. You can see me right here because I'm static. You can see my hand here because it's static. But if I'm going to rapidly sway my hand, if I go to a super high speed, you would stop seeing my hand here, which is very similar to what happens with these proteins. These are all structured, rigid core of the protein. But this part I've mentioned here is unstructured and flapping around. So there's no way you can see them in electron micrograph. So this is where these biochemical techniques come handy, where you can try to understand where the unstructured region also, interacts. So the final part of the talk. This is a totally different protein structure. So the protein shown here is called NS1A protein. It's an influenza A virus protein that has been shown to interact with this protein called CPSF30 in humans. And what CPSF30 does is, CPSF30 is a part of the mRNA polyadenylation machinery. And the influenza actually prevents the CPSF30 from carrying out its function. In other words, the influenza protein hijacks the PolyA tail process. If you look at my structure, we have a protein in pink called YTH1, and this YTH1 is analogous or similar to this human protein colored in orange. Since we already have a structure of the viral protein with the orange protein, what I did was I did a structural alignment, and the pink protein and the orange protein beautifully aligned well because they are supposed to be the human version and the yeast version, and they are similar. And next I removed the human protein. As you can see, the virus protein sits in between the yellow protein and the pink protein, and it physically makes it impossible for them to interact together. And also it sits in a very important region. Turns out that this junction here is where RNA binds to my complex. Imagine this. In the absence of this protein, my complex can freely bind RNA and add a PolyA tail in the cell, and everything is normal. But during viral infection, this protein is going to come and bind in this junction, and the normal RNA binding is affected, and PolyA tails may or may be added properly, therefore affecting the normal functioning of the cell. So one of the extrapolation from our structure is that we could propose a possible mechanism through which influenza virus hijacks a cell. This is a very good example where fundamental biological research led to something a bit more translational. I mean, it's still a very early model, a very early hypothesis, but it's leading us to somewhere translational. And that's why I think fundamental research is very important. In the long run, it might benefit a lot of people, even though at the moment it might not. So I'm going to propose a model for how CPF might function. So you might be familiar with these cartoons now. These are the individual proteins of the PolyA module and how they interact. Because of time limitations, I didn't talk about a few other complexes such as the cleavage factors and the other part of CPF. We do have information biochemically and biophysically showing where they interact with the PolyA module. And also we are trying to work on understanding where the RNA binds to the whole complex. In other words, we are thinking of an idea where this PolyA module acts as a central scaffold or a hub, bringing together this complex called CF1A, bringing together the RNA and the polymerase for effective PolyA tail addition. I'm sorry I didn't talk much about CF1A because of time limitation. And yeah, you have the PolyA tail addition once everything is in place. And this is just the summary of my talk. I was able to reconstitute a PolyA polymerase module in the test tube, and I showed that it can add a PolyA tail to an mRNA, and we've also able to determine the atomic resolution structure of the core part of the complex, and it was similar to some of the complex that has been already determined. And we also show that this interacts with few other factors, therefore leading to a model where it acts as a hub in this whole process. The next few things that we would like to do is to understand not just how the PolyA module works, but how the whole CPF works. And my final acknowledgement slides, I would like to thank Laurie Passmore here. She's been a wonderful supervisor, and she's taught me a lot, not only about how to do experiments, but also how to think in a broader picture and how to go about pursuing those experiments. And Anna Cassaniel here and myself work closely on this project to determine the structure, and Chris Hill, Anna, and myself and Manuel over here, we are all a part of what is called the team CPF, and all of us are focused on understanding the mechanism of how CPF works. And most of the work would not have been possible if not for a place like LMB, because you have a lot of central facilities which all of us can easily access and use, making things more collaborative and easier. I would also like to thank all the members of Passmore Lab who's been extremely supportive, my second supervisor, Kiyoshi Nagai, and Luca Pellegrini, my university supervisor, and my funding body, Gates Cambridge, for their funding. And thank you for listening to me, and please ask me a lot of questions.